Hi everyone, welcome to all. This video discuss about sterilization. Sterilization method have two types. First one physical method, second one chemical method. This video discuss about chemical sterilization method. In chemical sterilization, disinfection is process of killing of many microorganisms but not all microorganisms. Disinfection is the chemical method of sterilization. A chemical used for disinfection are called the disinfectant. Distribution of mode of action. There are three type of mode of action. First one distribution of cell membrane, second one modification of protein, third one modification of nucleic acid. In distribution of cell membrane have alcohol, detergent, phenol. Modification of protein have chlorine, iodine, heavy metal, hydrogen peroxide, aldehyde, ethyl oxide, acid. At last, modification of nucleic acid have the crystal violet and malachite green. Mode of action. First one, distribution of cell membrane. Alcohol. Ethanol is widely used to clean the skin before immunization or venipuncture. It acts mainly by disorganizing of liquid structure in membrane, but it denatures protein as well. Ethanol requires the presence of water of maximal activity. It form effective at 70% than 100%. The 70% ethanol often used an antiseptic to clean the skin prior to the venipuncture. Second one, detergent. Detergent of surface active agent composed of long chain lipid soluble hydrophobic portion and the polar hydrophilic group, which can be cation, anion, non-ionic group. Detergent interact with lipid in the cell membrane through the hydrophobic chain and the surrounding water through the polar group and distribute the membrane. Quaternary ammonium compound, example benzoalkonium chloride, or a cationic detergent widely used for skin anti-species. Third one, phenol. Phenol was a first disinfectant used in operating room by the Lister in 1860, but it rarely used in disinfectant today because it too caustic. Hexachlorophenone, which is biphenyl with a 6 chlorine atom, used in germicidal soap but conquer over the possible neurotoxicity. As the limited it is used. Another phenol derivative is a cerosol methylphenol, the active integrant in a lysol. Phenol not only damage the membrane but also denature the protein. Phenol not only membrane damage but denature also protein. Modification of protein. Chlorine. Chlorine is used as disinfectant to purify the water supply and to treat swimming pool. It also active component of hydrochloride, bleach, Clorox, which is used as disinfectant in the home and in hospital. Chlorine is powerful oxidizing agent that kill by cross-linking essential sulfodihydral group in a enzyme to form inactive disulfide. Chlorine we used hospital chemical agent. Next one, iodine. Iodine is most effective skin antiseptic used in medical practices and should be used prior to obtaining the blood culture and installing intravenous catheter because contamination with the skin flora such as Staphylococcus epidermis can be problem. Iodine sulfide in two form. Tincture of iodine iodophore. First one, tincture of iodine 2% of solution and potassium iodide is in the ethanol is used to prepare the skin prior to blood culture because tincture of iodine can be irritating to the skin. It should be removed with alcohol. Iodophore are complex of iodine with detergent that are frequently used to prepare the skin prior to surgery because they are less irritating than tincture of iodine. Iodine, like chlorine, it is an oxidant that inactivate sulfodihydral containing the enzyme. It also kind specifically to triosin reduced to the protein. Third one, heavy metal. Heavy metal, mercury, silver have the greatest antibacterial activity of heavy metal, the most widely used in medicine. They act by binding to sulfodihydral group They are blocking enzymatic activity. Thymosol, merthiolate and the merbomian mercurochrome, which contain mercury, used as skin antiseptic. Silver nitrate drop are useful in preventing gonococcal epithelioma nonotum. Sulfur Sulfur diazine used to prevent infection of burn wound. Hydrogen peroxide. Hydrogen peroxide used an antiseptic to clean wound and the disinfectant contact lens. Effectiveness is limited by organism ability to produce the catalyze and enzyme that degrade hydrogen peroxide. The bubble produced when peroxide is used and wound formed by the oxygen arising from the breakdown of H2O2 hydrogen peroxide by tissue catalase. Hydrogen peroxide is an oxidizing agent attack the sulfur-dihedral group, thereby inhibiting the enzymatic activity. Next, aldehyde. Formaldehyde, which is available as 37% solution in water. Formalin denature the protein and nucleic acid. Both protein and nucleic acid contain essential NH and OH group, which are the main site of alkylation by the hydromethyl group of formaldehyde. Glutaldehyde, which has two reactive aldehyde group, is the 10 times more effective than formaldehyde and is less toxic. In hospital, it is used in sterilized respiratory therapy equipment. Next, ethylene oxide. Ethylene oxide is a gas used extensively in hospital for sterilization of heat sensitive material such as surgical instrument and plastic. It is killed by alkylating both protein and nucleic acid. Hydroethyl group attack the reactive hydrogen atom or essential amino and the hydroxyl group. This also kill the protein and nucleic acid. Modification of protein lost. Acid. Strong acid and alkalis killed by the denaturing protein. Weak acid such as the benzoic acid, propanoic acid and citric acid are frequently used as food preservative because they are biostatic. The action of the acid is partially function of organic moiety, the benzonate as well as the low pH. Modification of nucleic acid is the last type of chemical sterilization method. Modification of nucleic acid. A variety of dye not only stain the microorganism but also inhibit this growth. They have two types: crystal violet, malachite green. First one, crystal violet. One of this crystal violet, genthanin violet, which is used in skin antiseptic. Second one, its action based on binding of positively charged dye molecule to the negatively charged phosphate group of the nucleic acid. Second one, malachite green. Malachite green is tri-phenylamine dye like crystal violet, component of Lewinson-Jensen medium, LJ medium, which is used to grow Mycobacterium tuberculosis, the special medium of Mycobacterium tuberculosis. The dye incubate growth of unwanted organism in the sputum during this 6-week incubation period. LJ medium like green color appearance, special type of Mycobacterium tuberculosis. That's all about chemical sterilization method. Thank you. Bye bye.